begin by thanking the organisers for the invitation to speak at this Asian Science Camp. I'm sure I speak for all of the lecturers when I say that it's a real privilege to be speaking to some of Asia's future scientific leaders. This is actually our first visit to Malaysia, and I'm really enjoying the culture, the landscape, and of course the food. So thank you very much for the wonderful hospitality. So during my talks this morning are centred on cancer. I want to begin with a brief historical overview of cancer and its causes, and then I want to introduce you to two very different cancer-causing genes and how they were discovered, and that's where our own scientific story fits into the picture. And because one of these genes proved to be a major regulator of cell death, I want to, in the third part of my talk, describe in more detail to you how cells decide to live or die. And this will set the scene for the next talk by Jerry Adams, who will tell you how deep molecular understanding of this life-death switch in cells has led to the development of very exciting and promising new class of cancer drugs. So these two talks fit together. So let's begin with an overview of cancer and its causes. Cancer is traditionally depicted as the crab, and it's one of humankind's most dreaded foes. And conquering cancer is an enormous task, because it's not just one disease, it's at least 100 diseases. Cancer is not a modern disease. In fact, the fossil records tell us that dinosaurs had it at least 70 to 80 million years ago, and our hominid ancestors had it at least 4 million years ago, and probably cancer has existed as long as multicellular organisms have evolved. Cancer was known to the ancients, and it was feared then just as it is feared now. Indeed, as early as 1600 BCE, the Egyptians recognized cancer as a disease, and of course, they blamed it on the gods. You know of Hippocrates, of course, known as the father of modern medicine. He was very aware, and this is a picture of Hippocrates here, he was very aware of different kinds of cancers, and it was he, in fact, who gave it the name cancer. He called it cancer because some cancers he was studying had very distended veins extending out from them, and he thought it looked like a crab. He dissected tumours, and he started to recognise the difference between normal and malignant cells, but he didn't have any idea of its cause. Well, it proved that he didn't have any idea, because at that time sickness was thought to be caused by an imbalance in the four body humours. And both Hippocrates and later the Roman physician Galen ascribed tumours to an imbalance in these body humours, and cancer in particular was due to too much black bile versus yellow bile or blood or phlegm. A very strange idea to us today. But can you believe it? This view about cancer was to hold for another 20 centuries. And why was that? Basically, because we didn't have enough scientific understanding about how our bodies develop and had very few scientific tools. It wasn't until, get this right in a minute. It wasn't until Percival Pott in 1775 in London that we realised that cancer could be an occupational disease. Because in 1775, he realised or postulated that there was a very high incidence of scrotal cancer amongst men who'd worked as chimney sweeps when they were boys might be due to the fact that they were exposed to so much soot. And then other associations began to emerge. In the mid-19th century, German pitchblende miners were noticed to have a high incidence of lung cancer. And the reason for that emerged when in the early 20th century, Marie Curie, that amazing woman, discovered that pitchblende contains radioactive elements, polonium and radium. And it was, of course, exposure to those radioactive elements that caused the disease in the miners. Almost as soon as tobacco was introduced into Europe, it was recognized to be a major health hazard. However, it was not so, and you can see that from this picture of Van Gogh of a skull smoking. So people suspected something to do with tobacco was causing lung cancer, but it was not until the exquisite and very detailed epidemiological studies of Dole and Pito that there was a causal link proven between smoking and lung cancer. And smoking still causes 30% of the cancer deaths in the US. You know, it's very hard for people who smoke to accept the notion that that will cause disease. And lung cancer is still one of the leading causes of cancer deaths in China, and I suspect all of Asia. And lest you be tempted to smoke yourselves, let me just show you this very graphic picture between the contrast between normal lungs and lungs that have been taken over by lung cancer. It's a dreadful disease. And you can see in the bottom right-hand corner this inexorable link between cigarette consumption and later the development of lung cancer. So do not smoke. So, what is cancer? To put it in a nutshell, cancer is an abnormal accumulation of cells within the body, could be anywhere within the body, due to a breakdown of normal cellular growth control. We call cancers by different names depending on where those cancers arise. For example, colon cancer arises in the colon, and leukemia and lymphoma, that we'll be talking quite a bit about in this lecture, arise in our blood and in our tissues where blood circulates. Okay, so the other thing that I want to tell you about is that cancer is, or a principle that I want you to take away, is that cancer is a multi-stage disease. And here that progression of cancer is depicted for colon cancer. So in the first part of this slide, at the top left-hand side, you'll see one of those cells is coloured differently because it's become altered. And it's lost normal growth control, a little bit of normal growth control. It might divide slightly more quickly than its neighbours. And that ends up creating a whole little patch of cells, the progeny of those cells, of abnormal cells. And then later again, one of those cells within that abnormal patch becomes changed still further and becomes more aggressive. And that goes on over the years until eventually you get this very aggressive, invasive cancer that breaks totally out of its normal cellular tissue boundaries and escapes into the bloodstream, gets carried to different organs, and that's what we call metastasis. And at that stage, this is a cancer which will doom this patient unless it's treated. So, what goes wrong? How does this happen? Before we consider that in some more detail, let's pause for a minute and consider how we develop as human beings. As you know, every tissue in our body is built from cells. Cells are the basic units of biology. And deep in the heart of every cell are your chromosomes, your genetic hard disk. And every cell in your body has exactly the same genetic hard disk. And of course, up in the left-hand corner, you'll see the very beginnings of life. Each one of us is built from a single cell which received half its chromosomes from the egg of our mother and a very similar set from the sperm of our father. And following the genetic instructions in these chromosomes, that single cell divided into 2 daughter cells, and these 2 daughter cells gave rise to 4 granddaughters, and so on, ad infinitum, until your body was complete. And along the way, the cells started specializing. Some became liver cells, some became neuronal cells, and each found its own position in the body plan. And let me now continue in the words of Bob Weinberg, a very insightful cancer researcher from the Whitehead Institute in Cambridge, Massachusetts. And I quote, "The 30 trillion cells of the normal healthy body live in a complex, interdependent condominium, regulating one another's proliferation. Indeed, normal cells reproduce only when instructed to do so by other cells in their vicinity. Such unceasing collaboration ensures that each tissue maintains a size and an architecture appropriate to the body's needs." Unquote. So how does this complex and highly regulated process, this amazing evolutionary result, go wrong and let cancers develop? Well, as we know today, cancer is basically a sickness, a malady of our genes, as Nobel laureate Mike Bishop so eloquently put it. Whoops. Let me go back now. So, cancer is caused by damage to the DNA in our chromosomes. That is what we call a mutation. Most mutations that occur in our cells have no consequences, and many of them are repaired by the very elaborate machinery that has evolved to repair our DNA. But occasionally, a mutation will occur that confers on that cell acquiring that mutation a growth or a survival advantage. And that gives the cell the potential to become a cancer cell. But just as I told you that cancer is a multistage disease, it takes more than one mutation to cause cancer. Multiple mutations are required for a cell to become fully malignant. And that's just as well for us, otherwise we would all be carrying cancer, we'd be all dead very young. And during this evolutionary selection process that goes on during cancer development, there's a selection for mutations that synergize with each other to create a much more malevolent and fast-growing cell. How do cells acquire oncogenic mutations? Well, some, in a few unlucky families, they're inherited from the germline. Some families carry genes that confer on that family a moderately increased risk of developing cancer later in their lives. But that's a very small number of the total number of cancers that occur in the world. Most occur by somatic mutation, that is, mutations that occur during our lifetime. And as I've told you, some of them are caused by external agents interacting with our body and changing our DNA. Exposure to tobacco smoke, or exposure to asbestos, or in the case of those chimney sweeps, soot. Some of us get mutations from things that we eat. But some of it, and probably most of it, is just plain bad luck. Because if you think of all the cell divisions that have to occur for our bodies to develop and maintain themselves during our lifetimes. Every time a cell divides and has to replicate its DNA, there's a chance that that process will go wrong. So many of the errors that we see in mutations are caused by errors in the replication of our DNA, errors in the natural process. Some cancers, there's a viral etiology. It's not that those viruses in humans cause the cancer, but they deregulate the normal processes of cellular division and proliferation, and they can push the cell towards becoming oncogenic or tumorigenic. Liver cancer is facilitated by infection with hepatitis B virus, and cervical carcinoma by HPV virus. So there is a viral etiology in some cancers, but not all. And we now know that there are two major classes of cancer mutations. There's activation of genes which encourage cells to divide too many times or survive when they shouldn't survive. We call those genes oncogenes, and you can think of that as the accelerator in the car being jammed on. And then there are another class of cancer-causing genes which normally suppress cell multiplication, which normally very strictly control it. And when those genes are damaged or lost, the brakes are disabled. So that also contributes to cancer. And these two kinds of gene families interact with each other in the cancer development process. What are these genes? Well, many of them, most of them, are components of the very complex circuitry that regulates a cell's response to its environment. The oncogenes, shown in green, are usually parts of biochemical pathways that stimulate cells to divide or survive. Whereas the tumor suppressor genes, shown in red, are inhibitory genes, inhibitory proteins in that process. So it's a balance of activation of oncogenes or loss of tumor suppressor genes which contributes to cancer development. Now, how were these genes discovered? And that is a really exciting, fascinating story that I could talk for many lectures in, so I can only just give you a glimpse about that. We started to uncover these genes from the end of the 70s into the 80s. It was an incredibly exciting period in cancer research, and it's still ongoing, and it's still very exciting. So let me just tell you a little bit about how some of these genes were discovered. Some of them were discovered by retroviruses. These are viruses that cause, many of them cause leukemias and lymphomas. The very first cancer-causing retrovirus was discovered in chickens by Peyton Roos, who's often considered the father of modern cancer research. And he found that he could take a tumor from a chicken, squish it up, filter it and get a filtered supernatant and inject that into healthy young chickens, and he could transmit their leukemia. So he realized that there was a virus there that was responsible for causing leukemia in the birds. But it took a very long time to understand what that virus was doing. And it wasn't until the end of the 70s that molecular biology came along and gene sequencing came along, and it was realized that there was one gene within that Ralph sarcoma virus that we call SARC, or SRC there indicated, that was responsible for the tumor-promoting activity of that retrovirus. And even more excitingly, and this led to a Nobel Prize to Mike Bishop, who I introduced you to before, and Harold Farmas, when they cloned the gene and they compared it to the normal cellular genome, very excitedly, they found that this cancer-causing gene from the virus was very similar, not identical, but very similar to a gene that's carried by all normal cells in your bodies and in mine, in normal chickens. That normal gene is called CSARC, the cellular sarc. And other retroviruses were quickly found to have viral counterparts, viral cancer-causing counterparts of normal genes. Now, those genes are not in our bodies to cause cancer. They're normally very important genes for regulating our cells. And it's only when they're expressed at a very high level, like they are, they're delivered by the retrovirus, or when they've been mutated, as many of them are, that they acquire the capacity to cause cancer. So the retroviruses gave us one group of oncogenes. Other oncogenes were discovered by chromosome abnormalities that were found in leukemias and lymphomas. And here I've shown you a picture. On the right you'll see a leukemia, a very nasty acute lymphocytic leukemia with far too many white cells, which are stained blue in this side, compared to a normal blood smear on the left. So very aggressive leukemia. And so this is where Jerry and I got into the picture. Towards the end of, the beginning of the 80s, we started reading in this new field. And we basically dropped what we were doing. We became so excited by these discoveries that we changed fields altogether and started working on these chromosome abnormalities in leukemias and lymphomas. So I'll tell you a little bit about our own story in these next few slides. So let's go back for a minute and ask, so I said that many leukemias, I haven't told you that yet, well, many leukemias have very specific chromosome abnormalities that we call translocations. What's a translocation? Basically, a translocation is when two non-homologous chromosomes break and get joined together with the wrong chromosome. So you get two hybrid chromosomes in that cell, and I've shown them as yellow and red. Now, the important and big clue was that different kinds of leukemias had different chromosome abnormalities, different chromosome translocations, but every patient with a particular leukemia carried identical chromosome translocations. Now, why was that? And there was a lot of argument at the time. Were these abnormalities a cause of the cancer, or were they a consequence of the cancer? And why were they so often associated with leukemias and lymphomas? Well, Jerry and I had been working on antibody genes, and we knew that lymphocytes have to undertake a very fascinating rearrangement process in order to be able to synthesize antibodies. Basically, you don't need to understand the full process, but you do need to understand that the antibody genes are encoded in our germline in bits and pieces, and parts have to be joined together to make a functional antibody gene. So I've shown that process here. One of the V elements, one of the D elements, and one of the J elements get joined together to make a complete functional variable region of the antibody gene. So all normal lymphocytes have to undergo this DNA rearrangement process. And what we wondered at that time was, could chromosome translocations represent an antibody gene rearrangement that had gone wrong? And basically, instead of the intrachromosomal DNA rearrangement, there was an interchromosomal rearrangement. Could an oncogene have been joined into the antibody gene locus? And you know, that seemed like a scientific dream at the time. But that dream came true. Sometimes dreams do come true. And we started working on a lymphoma called Burkitt's lymphoma, which has this 814 translocation. And just at about that time, it was realized that the break point on chromosome 14 is where the antibody heavy chain gene locus is. But what was coming in, what was being joined to it from chromosome 8? Well, what we did was to use part of the antibody gene to track down unusual gene rearrangements of that gene in those cells. And what we found was that, just as we had predicted, the antibody gene locus had been joined to a cancer-provoking gene that was already known, a gene called MYC. It had already been identified as a gene in a retrovirus. And what happens when MYC gets joined to the antibody gene locus is that MYC is given a really strong signal to always stay on. So cells keep dividing when they should stop. Now it seemed at that point, when we and independently three labs in the US found that MYC was at that translocation breakpoint, that MYC was the cause of this Burkitt's lymphoma. But it was a guilt by association. We wanted to prove it directly, so we turned to a very new, at that time, now very routine technique called transgenesis, whereby what you do is inject the DNA into a fertilized egg, and that fertilized egg is implanted into a pseudo-pregnant mouse, and every cell in the body of the pup that is born from that injected egg carries the extra gene that you put in. And in our case, of course, what we put in was a MYC gene joined to this antibody gene locus. And very exciting, and we had to do this in collaboration with scientists in the US because nobody in Australia could carry out this transgenesis at the time. It had only just been developed. And we collaborated, and there was this amazing phone call that we got from our colleagues in the US when they said, the mice are coming down with lymphomas. And I will never forget that phone call, that transatlantic phone call. Trans-Pacific, sorry. Anyway, you can see the mice, we call them MYC mice. Every single MYC transgenic mouse develops a B lymphoma, and you can see those lumps on their sides. And they've been an incredibly powerful model to study how this process occurs. Basically, what we found is that even that very powerful MYC gene from the translocation is not sufficient to fully transform the cells. So what happens in young mice is they've got abnormal cells, but if you transplant them into another mice, they don't cause cancer. But if you let the mouse age, those cells develop other mutations, and eventually you get a tumour that is transplantable. So again, an example of how cancer develops in stages and how you need multiple genetic changes to cause cancer. Okay, so these mice and that translocation provided a paradigm to identify many other oncogenes activated in leukemias and lymphomas. And the next one we turned to was human follicular lymphoma, which has a different chromosome translocation, a 14-18 translocation. We didn't clone this one, that was cloned by other labs in the US, but our lab discovered what that gene does, because that gene, when it was cloned, was different to any other gene that had been isolated at the time. Its structure, its sequence gave no clues as to its function until this experiment carried out by David Bowe in our lab at the time, who was a PhD student at that time. So that tells you you can do amazing things when you're a PhD student. You don't have to be a mature scientist. You can make groundbreaking discoveries even when you're a student. What David did was he took a retrovirus, he made an artificial retrovirus carrying this BCL2 gene, and he infected cell lines that were totally dependent for their growth and division on having a cytokine called IL-3 in their medium. So here are the cells on the left growing happily with they've got cytokine. If you take out that cytokine, the cells will rapidly die. But the cells that had the BCL2 retrovirus in them, they did not die when he took out the cytokine. Instead, they stayed alive, and they stayed alive for weeks. They did not divide anymore, but they stayed healthy. So this was a very different oncogene, very different from MYC. Instead of encouraging cells to proliferate, it's an oncogene that's encouraging cells to divide under conditions in which they should die. Now we made transgenic mice with BCL2, and we crossed them to our MYC transgenic mice. So just with a BCL2 transgene, the mice are not highly susceptible, that's in blue, are not highly susceptible to cancer. Whereas the MYC mice, all are dead with a lymphoma by 12 months of age, as I told you. But their progeny that carry both the BCL2 oncogene and the MYC oncogene develop lymphomas much faster. And why are they doing that? It's because, as other scientists first found out, if you express MYC, when cytokines are limiting in amounts, MYC provokes cell death. And you can see that in the next experiment. If you take out B lymphoid cells from those mice, in black is shown the rate of death in culture of normal B lymphoid cells from a normal mouse. But from the MYC transgenic mice, those cells divide much faster. But look up there in orange. Cells from a mouse that's carrying the MYC and the BCL2 gene are very resistant to cell death, just as resistant as cells that just express the BCL2 gene. Now those experiments were groundbreaking. They totally changed the rules, and we now know that blocking cell death is a central step in the development of probably all tumours. Okay, so that leads me to the final part of my talk, which is the very important process that we've been studying for the last 30 years, because it's such an important process. This process of cell death by apoptosis. How does it occur, and what's its role? Well, apoptosis is absolutely vital for health. It plays an incredibly important role during development for sculpting embryonic tissues. For example, the interdigital cells during our development die, and so we have digits, whereas they don't die for a duck, and a duck can paddle. It's very important, this process of cell death, in ensuring that we have the right balance of cells in our tissues, so we don't keep growing our tissues. And it's particularly important in regulating the number of cells in tissues like the immune system or the gut, where a lot of cells are born every day, and an equivalent number should die every day as well. And it's genetically programmed. And here is a cartoon of the process. So that's a normal cell on the far left. Cells during apoptosis shrink, their DNA is cleaved, the cells bubble away, bleb, and they eventually break up into little subcellular bodies that are rapidly engulfed by neighbouring macrophages. It's a very fast process. The cell, from life to death, it takes about 3 minutes. It's very clean, and that's very important. Because these cell bodies don't break open, they don't release their contents, and so there's no inflammatory response. This is a picture on the left of a normal lymphocyte shown by scanning EM. At the top is the normal lymphocyte, and this is what a cell looks like when it's undergoing this rapid blebbing and breaking up into apoptotic bodies. And over the last 30 years, we've worked out, this is a much bigger we than just Jerry and me, worked out, scientists all around the world involved in this. I've told you the very first gene that was identified that controls this process that came from cancer genetics, and that's BCL2, but other clues came from developmental genetics and biochemistry, really elegant biochemistry. And I'll tell you a little bit about that. So Bob Horvitz worked in this nematode worm, and he had mutagenized the worm, and he realized that, well, it is known that exactly 131 cells out of 1,090 die during the development of this worm. And he found 3 genes that promote more cell deaths during development, and another gene that protects against cell death during development. And he called them, these genes, the names that you see on the right. Now, when these genes were cloned, it turned out that SED9, the one that protects against cell death in the worm, is homologous to BCL2. And the next clue came when SED3 was cloned, and it was realized to be homologous to enzymes in mammalian cells called caspases, cysteine proteases that cleave after aspartic residues. Now, these are very dangerous proteins, so they're normally held in check. They're normally synthesized as inactive zymogens. And only when they're cleaved, and you can see in the top left where those little arrows are, between those subunits will they become activated. We needn't go into the details about that, except to say that there are many caspases and their specificities vary. And they can cleave between them hundreds of vital proteins in the cell, and that's what dooms the cell to death. So they're the executioners. Okay, back to this slide of C. elegans. We've worked out what SED9 and SED3 are in mammalian cells. What about this SED4? That was worked out by really elegant biochemistry. Lots of people were looking for the mammalian homolog of SED4, and it turned out to be a scaffold protein that is activated when cytochrome C leaks out of mitochondria. It acts as a scaffold for the aggregation and activation of one of these caspases, procaspase 9. And when procaspase 9 activates itself, it then recruits and activates its downstream caspases, such as caspase 3. So here is this wheel of death. When APAF1, the equivalent of SED4, mammalian equivalent of SED4, is active, it has aggregated all these caspase 9 units onto this apoptosome, which we call the wheel of death, and that sets off this train of caspase activation. So here is the process summarized in a cartoon. When mitochondria become leaky and cytochrome C comes out, that activates APAF1, the apoptosome forms, caspase 9 is activated, and then subsequently caspase 3, 6, and 7. So there's this cascade of caspase activation. So let's come back to the really important point for us. How does BCL2 regulate this process that activates caspases? Well, it turned out that BCL2 had many relations, about 20 of them. Some of them, like BCL2, block cell death, and you don't need to remember the names, but the blue ones block cell death. But then there are 2 other families that, instead of blocking cell death, activate cell death. The orange ones, particularly BAX and BAC, are very similar in sequence and structure to BCL2, but nevertheless they activate cell death. And then there are more distant cousins, which we call BH3-only proteins, because they only contain one region of similarity to BCL2. They also cause cell death. Those proteins assemble on the mitochondrial outer membrane, and they do battle with each other. The pro-life and the pro-death members of the family determine whether or not the mitochondrial membrane retains its integrity. And the survival of all cells depends on the expression of at least one of these guardians. That's a very important principle. Another very important principle from this slide is that it takes both the Bax subfamily and the BH3 only proteins to cause cell death. Now, how does that happen? Well, in a healthy cell, there's lots of BCL2 and its colleagues, and they block the activation of Bax and Bac. They prevent cell death, and so the cell survives. But in a stressed cell, signals come in that activate the expression of the BH3 only proteins, and they bind to the pro-survival proteins and prevent their activity, prevent them from blocking the activation of Bax. And that can lead to the damage to the mitochondria, which releases the cytochrome C. Some of the BH3 only proteins also directly bind to Bax and Bac and activate them and cause cell death. So then when that process is happening, if the pro-death proteins overwhelm the pro-survival protein, then the cell commits cell suicide by activation of the caspases. This whole process of the interaction between these three kinds of family members is like a life-death switch, and it's understanding in intimate detail how that switch operates, which has been really important to develop for the development of new drugs, as I'll tell you. But let me just tell you a couple of other things first. The BH3 only proteins, of course, are really dangerous. So they're not expressed except under very high conditions of stress. And different stresses and different cytotoxic agents activate different BH3 only proteins. For example, if cells acquire DNA damage, that activates a transcription factor called p53, which activates the expression of two of the BH3 only proteins, fumarinoxa, and others are activated by different signals. So this is the critical piece of information that you need to keep in mind. Is what happens when a BH3 only protein binds to a pro-survival protein. Well, the pro-survival protein is shown in blue. It's a globular protein, but on it, the structural biologist taught us that on its surface it has this hydrophobic groove. And into this hydrophobic groove is where this conserved domain, the BH3 domain of a BH3 only protein, binds. It binds very tightly, and that very tight binding is what inactivates the pro-survival proteins and prevents them from inhibiting Bax and Bac. So that is the critical binding reaction. How are Bac and Bacs activated by BH3 only proteins? Well, basically the same way. The BH3 domain of a BH3 only protein binds to the groove that Bax and Bac have. And that enables, so the difference between the pro-survivals and Bax and Bac is that Bax and Bac are much more unstable proteins. And when a BH3 only protein binds, that makes this huge conformational change, which enables Bax and Bac to go on and form dimers, homodimers, and they form oligomers, and somehow, in a way that we don't understand yet, and this is the holy grail in the field at the moment, that creates pores in the mitochondrial membrane, and cytochrome C escapes. Now this is my last slide. This is the introduction to Jerry's slide. So I've told you that this switch is comprised of three classes, three subclasses of the BCL2 family, the BH3 only proteins, which bind to the pro-survival proteins and neutralize them, and Bax and Bac, which activate, which create holes in the membrane of the mitochondria and activate the caspases. Tumour cells, I've also told you, resist apoptosis, and they do that by either overexpression of BCL2 or its colleagues, or they have defects in the induction of the expression of BH3-only proteins. But most of the cellular machinery of cell death is intact in every cell, in every cancer cell. So we, and of course many other labs and the industries out there, reason that we should be able to find small molecules that bind to that hydrophobic groove in the pro-survival proteins and mimic BH3-only proteins, overwhelm the pro-survival proteins, and cause the activation, therefore, of BAX and the death of the tumour cell. So that's the story that you'll hear in the next lecture from Jerry. I want to just finish by introducing you to our institute in Melbourne, the Walter and Eliza Hall Institute. It's now over 100 years old. It focuses on cancer and immunity and infectious diseases. There's lots of exciting science done there. It's a wonderful place, and I hope that some of you someday come here to do your own PhD studies. It's very multicultural and very international, and we would certainly welcome you coming to join us in Melbourne. So thank you for your attention.